request lagu Pak Hari.
Ya Bu.
Boleh request lagu ya?
Boleh.
Bagus
Tapi
banget.
jangan yang copyright.
Ya.
Iya, kalau copyright nanti channel YouTube-nya kasih ya. Pentung. Bapak Ibu peserta yang ingin saling menyapa, dipersilahkan. Kalau ada kenalannya mungkin bisa sekalian silaturahim.
Ada Bu Lucy, saya lihat Bu Lucy Unpad. Lucy ada ya?
Ada Bu, ini Bu Asti yang bicara. Ya, ada Bu Asti. Terima kasih sudah bisa hadir di sini.
Ya. Bu Lucy ini bakteriologis dari Pajajaran.
Iya, makanya penyakit darah itu sangat menarik buat saya, Bu Asti.
Ya. Mudah-mudahan bisa tukar pengalaman ya Bu.
Iya, betul Bu Asti.
Pak Hari, silakan.
Sudah bisa dimulai?
Sudah.
Oke, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. My name is Hari Priwiratama. I will be your host in today's session. For those who don't know me, I am a second semester doctoral student in the study program of Phytopathology, Department of Plant Protection, IPB University. It is my pleasure to welcome you all to the guest lecture series 14 in the topic of plant pathogenic bacteria. Just so you know that the participant in this lecture is not only students enrolled in plant pathogenic bacteria course in IPB University, but also academicians from other universities, research institutes, government agencies, as well as professionals. This guest lecture also streamed live in our YouTube channel. First of all, let us thank to the God Almighty for His blessing to make this guest lecture possible. Today's session will have a special lecture covering the information on the biology and epidemiology of banana blood disease. This is a very interesting topic as Indonesia is known as one of the center of origins for banana, and the fact that the banana blood disease was first reported in Indonesia, to be exact in Sulawesi Island back in 1900. As the guest lecturer in today's session, I noticed that many of her research in banana blood disease also was conducted in Indonesia. Ladies and gentlemen, before proceeding to the agenda, I would like to remind you all to turn off your microphone during the session. And if you have questions during the lecture, please kindly write it down in the chat column. Bapak dan Ibu, untuk pertanyaan dapat juga disampaikan dalam bahasa Indonesia untuk nanti akan diterjemahkan oleh moderator. Last but not least, the link of the end of the lecture. So please ensure to follow the lecture until finish. And now we will have a welcoming remark by Professor Sri Hendrastuti Hidayat, or usually called Bu Asti. Bu Asti is the head of Phytopathology Division in the Department of Plant Protection, IPB University. I would like to invite Bu Asti to turn on the microphone, please. Bu Asti, how are you today?
Oh, thanks, Hari. I'm fine. Is it?
Yes, Bu Asti.
Okay.
Are you ready?
Sure.
Okay, Bu Asti, please, the Zoom is yours.
Oke, thank you Hari. Terima kasih Hari sebagai MC pada hari ini. Selamat siang Bapak-bapak, Ibu-ibu, dan saudara-saudara semuanya. Good afternoon everybody. Assalamualaikum warahmatullahi wabarakatuh. Senang sekali hari ini kami dapat menyelenggarakan acara guest lecture yang ke-14. Acara ini merupakan bagian kegiatan dari Departemen Proteksi Tanaman IPB dalam rangka meningkatkan pengetahuan dan wawasan mahasiswa IPB tentang topik-topik ilmiah tertentu, juga dalam rangka berbagi atau sharing pengetahuan serta pengalaman dengan rekan-rekan akademisi, peneliti, dan praktisi di berbagai institusi, terutama di aspek bidang proteksi tanaman. Atas nama Ketua Departemen Proteksi Tanaman, kami menyampaikan rasa terima kasih dan penghargaan atas partisipasi dan kehadiran Bapak-bapak, Ibu-ibu, dan saudara-saudara sekalian pada hari ini. Today, as our MC told you earlier, we have a special speaker, an expert in banana blood disease, especially for the case in Indonesia. Thanks to our old friend, Jane Ray. Hi, Jane. Jane is now affiliated with Northern Territory Government of Australia in Darwin. So this lecture is held, in two countries, Indonesia and Australia. It is our pleasure to have Jane as our speaker. I think most of us are very familiar with blood disease of banana because, as mentioned earlier by Hari, the disease was first reported from Selayar Island in South Sulawesi, but it is now widely spread across the country and even to banana production countries in Southeast Asia and maybe more. I'm not going to spoil too much about this disease because we will hear a thorough and comprehensive story directly from the expert. So Bapak dan Ibu serta saudara-saudara sekalian, saya haturkan selamat mengikuti kuliah ini dan semoga akan memberikan banyak manfaat bagi kita semua. Wassalamualaikum warahmatullahi wabarakatuh. Saya kembalikan kepada Hari sebagai MC. Terima kasih.
Thank you, Bu Asti, for warm welcoming and also introducing the guest lecture series today. Ladies and gentlemen, before proceeding to the lecture, we will have a photo session. Please kindly toggle on your camera. And I noticed that there are several panels in my view, at least. So there will be a photo session. Please stand still until our team finish capturing all slides. Let's begin with the first. Let's begin with the first panel. 1, 2, 3. Okay, the second panel. 1, 2, 3. And the last panel. 1, 2, 3. Thank you for your patience in photo session. All participants, within a few minutes, we will proceed with the lecture. The session itself will be chaired by Mrs. Sari Nururita, or we usually call in 3 simple letters, Bu Ita, I-T-N-A. But this is not codon, of course. Bu Ita is one of lecturer in plant virology, the Department of Plant Protection, IPB University. She earned her bachelor and master degree in IPB University, and currently she is at her final phase of her PhD in the field of plant virology in the University of Queensland, Australia, which is expected to be finished by June this year. So without further ado, I would like to welcome Bu Ita to toggle on the microphone, please.
Mas Hari, thanks
Yeah.
so much for the introducing me.
It is good to see you.
Good to see you again.
Yeah. We hope that you can finish by June this 2022, and we hope that everything goes well as planned.
Thank you. Amin.
So without further ado, Bu Ita, please, the Zoom is yours.
Thanks, Mas Hari, for housekeeping this talk, and Bu Asti as well for welcoming Mark in this guest lecture series. Good afternoon, everyone. Today we'll have a special guest who is Miss Jane Ray, which is also my good friend at UQ. Before we started with her talk, I would like to introduce her. Jane Ray is a senior plant pathologist based in Darwin, Australia, with the Northern Territory government. She has over 20 years of experience working with plant disease and recently completed her PhD studies at the University of Queensland on the biology and epidemiology of banana blood disease. Congrats, Jane. Prior to this, Jane was a senior plant pathologist based in Darwin, working for the Northern Australia Quarantine Strategy, a program of the Australian federal government. For over 12 years, Jane conducted surveys along Australia's northern coastline and neighboring countries to monitor for early signs of new disease, and was involved in various overseas capacity building and training programs. She will be presenting a seminar on her PhD studies, biology and epidemiology of banana blood disease. I believe everyone looking forward of your talk, Jane. So without, before I started and welcoming Jane, I would like to friendly reminder, after Jane finished her talk, we will have a short time for a quiz. So I will read 2 questions that we hope participants can answer, and who comes first will have a gift, a special gift for us. And for the participants, we provided for winners, which came from Zoom and YouTube channels. Okay, and okay, hi Jane, how are you?
Very well, thank you. It's lovely
Good to see you again. Yeah.
Thanks for inviting me today.
No worries. Thanks so much for sharing with us. Hopefully, students and participants can have a lot of inspiring story and knowledge for your presentations. Okay, Jen. I think time is yours, and for participants, please don't forget to mute your Zoom during the presentation. Thank you.
Okay, I'll move to share my screen. Can I just check with Ita? Can you only see my presentation slide now?
Yes, perfect.
Selamat datang. Terima kasih telah menghadiri seminar saya. Hari ini saya akan mempresentasikan proyek penelitian 4 tahun saya untuk disertasi saya. Saya minta maaf untuk selanjutnya saya akan presentasi dalam bahasa Inggris. Today I'll be presenting biology and epidemiology of blood disease. Now, epidemiology is the scientific study of disease, its detection, spread, and its control. This work is the result of a collaboration between Gadjah Mada University and the University of Queensland, Australia. I now work as a senior plant pathologist for the Northern Territory Government and am based here in Darwin, Australia. Now, after I've finished my presentation, I'm happy to answer any of your questions. So I'd first like to thank the Department of Plant Protection, IPB University, for the opportunity to present my research today. I'd also like to thank the organising committee and Professor Asti and my colleague Sari Nuralita. Thank you very much for the opportunity to present today. And I'd like to thank Hari Prihawatna for your introduction today. And I apologize now for my pronunciation. So in regard to this research, I would like to acknowledge that this work was conducted as part of a team of people, and I'm very grateful for all of their input and support throughout this 4-year project. I'd particularly like to acknowledge the contribution of my supervisory team, in particular, the contribution of my principal advisor, Professor Andre Drenth, and Professor Siti Sobandia from Gadjah Mada University. Now, these two people made this project possible, and they made this project a success. And I mean, I'm very, very grateful for their input and support throughout this whole project. Now, of course, I'd also like to thank all of our funding providers and people who sponsored this work. So blood disease causes substantial crop losses in Indonesia and Malaysia. Indonesia alone produces around 7 million tonnes of bananas a year. And bananas are an important staple in this part of the world, as I'm sure you all know. And very important because about 50% of the population are smallhold farmers, many of whom live below the poverty line. Now, the Kepok banana variety is very, very popular in your part of the world, and it's highly susceptible to banana blood disease. Now, blood disease is a bacterial wilt that affects bananas. It's caused by Ralstonia syzygiae subspecies celebens. Now, the disease causes characteristic symptoms. The fruit pulp rots, there's red-brown vascular discoloration, and the leaves wilt and become necrotic. And certainly, once bananas or banana plants are infected with this bacteria, the fruit is completely inedible. Now, local transmission of blood disease is believed to be predominantly through insects mechanically transferring the bacteria on their bodies from diseased to healthy male banana bells. Tools, soil, and water have also been implicated in disease spread, whilst long-distance spread is hypothesized to be through the movement of contaminated planting materials. So these are all really great theories, but they're actually assumptions. There's very little data to support these theories. And a lot of these theories are based on field observations and on other species of bacteria that cause wilt on banana. And I'm talking here about mycobacterial wilt, which is caused by a related bacteria, Ralstonia solanacearum, and Xanthomonas wilt. Now, these two banana diseases have had quite a lot of biological work done on them over the years, but they're actually completely different species from the causal agent of blood disease, the Ralstonia syzygia that I was talking about. And they've evolved in completely different parts of the world. So in reality, there's actually just so much we don't know about the epidemiology and biology of blood disease. And this is really basic research, but and this leads me on to my research. Basic, by basic, I mean fundamental. We call it fundamental research. Now, due to the past limited geographic dispersal of blood disease, relatively little research has been conducted on its biology and epidemiology. So with this in mind, we developed 4 broad research areas. So these are actually the 4 chapters from my PhD. So first we look at the history of the disease and its current distribution. Then we look at the mode of infection and the location of the inflorescence infection courts. We then move to transmission and dispersal of the pathogen. Lastly, we look at diagnostics. So here we've developed and validated specific PCR diagnostics. So each of these 4 parts that I've moved through, each of them is a publication, and I can make those publications available to you at a later date. So the first part of my research, so here we look at geographic expansion of blood disease in Southeast Asia. So we first developed specific research questions for this chapter. So what is the historic and current disease distribution? Are there specific patterns in the way the disease disperses? Which banana varieties are susceptible in the field? And what is the identity of the isolates? And are they actually pathogenic on banana? So here we do Kopp's postulates. Now, to answer these research questions, we first conducted an extensive literature review. Now, every good study, as you know, starts with an extensive literature review. Then we did surveys across Indonesia to determine the distribution of blood disease. And we collected isolates from the diseased plants that we saw across Indonesia. Now, when we isolated the bacteria, we did it on a media called tetrazolium chloride media. And on this media, the blood disease is very distinctive. It has a red center and a white margin, and it's quite slow growing. And we also checked that the isolates were gram negative and that they were oxidase positive. Then we moved to check the isolate pathogenicity through Kopp's postulates to prove disease causality. And then we verified the identity of those isolates using specific molecular techniques. So we used specific primers and we sequenced a portion of the EGL gene to confirm the identity of the isolates. So what did we actually find? So this slide is a combination of the literature review and the survey results. It's the history and distribution and the pattern of spread. And I'm just going to talk you through the work that we did. So blood disease was first observed on a small island south of Sulawesi just over 100 years ago. Now, blood disease completely destroyed the existing commercial plantations on this island, Kayudi Island, and it caused those plantations to be abandoned. So then we found, then they found that blood disease was present in southern Sulawesi. And so we're talking about 100 years ago now. And it was found endemic in this part of the world. Now, at this point in time, the disease was not known from anywhere else in Indonesia nor anywhere else in the world. And so they put the local government at the time put a quarantine in place, preventing the movement of banana material from this region. And this quarantine was incredibly successful. For over 60 years, there were no further detections of blood disease. Blood disease was almost completely forgotten until in 1987, it showed up in West Java. And from here, it rapidly spread over the next 30 years and has recently showed up in Peninsular Malaysia, more recently Sumba. Blood disease, when it arrives to a new area, crop losses certainly approach 100%. It completely wipes out the crop. It's certainly a pathogen on the move, and it's certainly an emerging threat to banana cultivation in this part of the world and beyond. So now we look at this case study on the island of Sumba to demonstrate dispersal at the local level. So BDB was first noticed on the island of Sumba, Indonesia, in about 2011, just on the west side of the island up there. Now, at this time, or before 2011, there was a thriving banana industry in Sumba, and the disease just swept through the island in 4 years, becoming endemic. And this resulted in a total collapse of the banana industry in Sumba. The banana barges don't even come to this area anymore to pick up the bananas to take them to market. So now the pattern of short distance or local spread radiated rapidly from a single infection source. Now this pattern of radiation is suggestive of dispersal by insects, tools, water, or contaminated soil. Now, during the surveys, we confirmed blood disease in a total of 154 banana plants. Now, it was most commonly found in the Kepok banana. We got 121 isolates out of Kepok. But Kepok is also the most commonly grown banana over there, as you know. But we also collected blood disease from a wide variety of bananas across 4 different species. Excuse me. Musa paradisica, Musa acuminata, Musa balbisiana, and Musa textilis. The Cavendish banana is also susceptible, and this is important because obviously the Cavendish banana is incredibly popular in the rest of the world. And there's no known resistance within the Musa. It has a wide host range. So we performed Koch's postulates on a set of the isolates that we'd collected. Now, this is super basic, but super important concept in plant pathology. So as I'm sure you all are aware, we isolate the bacteria from symptomatic plants in the field. We inoculate test plants with the bacteria. We also have a control that we inoculate with water, and we obtain symptoms. We re-isolate the same organism. This is super important because it proves that the isolates that we collected in the field are pathogenic, and it proves that the causality of disease. So we then moved to identify the isolates as Ralstonia syzygiae subspecies celebensis using the specific PCR and sequencing of part of the EGL gene. So in our study of the distribution of blood disease, we have determined its current distribution, its pattern of distribution, the pathogenicity and identity of the isolates, and that it has a wide host range in the Musa. The disease has recently expanded its geographic range, and it's certainly an emerging threat to banana cultivation in the region. Now, if you'd like more information on this section or more details, this work has already been published in Plant Disease, so geographic expansion of blood disease in Southeast Asia. Now moving on to the next section. So for this chapter, we really wanted to understand inflorescence infection biology. That is where and how infection by blood disease actually occurs. So this is really fundamental research. Now, Ivan Budenhagen theorized that infection predominantly occurs through open xylem at the male bell. Down here, so this is the male bell. But this theory was actually not supported by data. There was no data behind this theory. And it's a great theory. And that leads us on to the specific research questions for this chapter. So firstly, we want to establish if there was a relationship between inoculum concentration and disease development. Some really basic biology. Does infection occur through the male and female parts of the inflorescence? Now, this is quite important for controlling the disease. And does infection occur through open xylem? Now, blood disease is exotic to Australia. So to answer our research questions, we set up a field trial in Indonesia, in Bantul, Yogyakarta. So the arrow kind of points to that region. So we planted about 160 kepok and 160 Cavendish plants. And when they were flowering, I actually moved over to Indonesia for about... I moved over to Indonesia for about 9 months to complete experiments on full-grown and potted banana plants. Now, this section of work actually consists of several different experiments that I'll talk you through. Now, you can see from my, one of the things we did was when the bunch emerged from the banana plant, we actually sealed them or bagged them in cloth bags. And this was to make sure that up here, they're quite tall bananas. This was to make sure that we didn't get any unplanned infection in the field trial. Unplanned infection of blood disease, because blood disease is endemic in this part of Indo. Now, I just thought I'd talk through some of the challenges as well to doing fundamental research or working on experiments on whole plants in a field trial. Now, to work at the field location, I needed to create a field laboratory because Bantul is actually quite a distance from Gajah Mada University. So I made a sterile cabinet so I could make inoculum and do isolations. Now I built, you can see it on the top left here. So I built this sterile cabinet out of a plastic box and some perspex. I used a lot of ethanol and I had a spirit burner and I worked under the spirit burner. And I was able to work to keep everything completely sterile when I worked in that cabinet. So it worked very well for me. So that was at the field laboratory. And there were some other challenges to do with doing field work, which I hadn't quite expected. But so one of the first challenges was when the bananas started flowering, the bells started going missing. And we had to actually put, and I need those bells for my inoculation experiments to find out what part of the inflorescence is an infection port. So we had to stop the villagers from removing the banana bells. And so we put up a big sign across the field. I'm not sure if you can read that or not. And the other things we had to control, we controlled banana bunchy top was problematic in the field trial, and banana scab moth. And down here, so banana scab moth, and that was causing again, it was causing frass to occur at the sites that I wanted to inoculate. So we really needed to control these things before we could even move to doing our experimental work. And then, of course, the dreaded COVID-19 came along and I had to leave the trials a bit early, but we still got there. Now I'll move back to the experiment. So this was the first experiment and we started with the basics. So I first wanted to know if the concentration of inoculum affects the development of disease. So I inoculated potted Kepok with the bacterium diluted from 10 to the 10 colony forming units down to 10 to the 2 colony forming units per mil. And I did a serial dilution. And I inoculated the stem of the plants by injecting them. So this was replicated 3 times and the control was inoculated with water. And we found that all dilutions from 10 to the 10 down to 10 to the 2 caused disease development from in all of the replicates after 18 days, although symptom development was a little slower below 10 to the 4 colony forming units per mil. So this means that this actually was a bit surprising for me. It means that only one or a few bacterial cells are required to enter the plant to initiate infection. And once infection is initiated, the same set of symptoms develop, maybe a little bit later if it's a very low inoculum dose, but eventually the plant dies. So in this experiment, we wanted to know which parts of the banana inflorescence are susceptible to infection by blood disease. So for this experiment, we used those field-grown banana plants. And I inoculated freshly revealed plant parts of female flowers, Malbel braxcars, Malbel flower cushions, snapped rashes, and deflowered fingers. And this was replicated at least 3 times. The whole experiment was replicated at a later date, and it was done in both Kepok and Cavendish plants. It was a huge amount of plants for this experiment. And we assessed whether or not those plant parts are infection courts through symptom development. And I also isolated the bacteria and checked that it was blood disease. Now you can see from my table here that all of the plant parts were susceptible to infection with the exception of the maturing fingers. But what's really important here is that the male and the female parts of the inflorescence are susceptible to blood disease. Now we move along to the next experiment, and this one again is in full-grown banana plants. So in this experiment, we wanted to determine if the xylem are open and does infection occur through open xylem. So we conducted a series of experiments using blue agricultural dye to test this open xylem theory. So dye was externally applied to freshly revealed plant parts. So I applied dye to the male flower cushion, the male braxcar, the female braxcar, and the snapped rashes. And then I monitored the plants after 24 hours. I harvested the plants, I chopped them down and had a look. And I was very, very surprised. But when you apply dye to these external plant parts, it just gets sucked inside the plant. And you can see from my images here, that's the blue dye going up through here and the blue dye coming up from the snapped rashes here. And I was super surprised. So now the dye just being sucked inside the plant like this provides really strong evidence that yes, the xylem are open, but not just that the xylem are open, but those plant parts are actually subject to a really strong negative hydrostatic gradient. So if something lands on them, then like a bacterium, it just gets sucked inside the xylem of the plant. Now, these plant parts with open xylem were also shown to be infection courts in the previous experiments. And all of this data together strongly supports the theory that infection occurs through open xylem. So we've shown that infection of the fruit can occur through the pedicel. which is the end that attaches the fruit to the tree or to the pseudostem, but can it occur through open xylem at the flower end of the fruit? So here we use that same blue agricultural dye, and I applied it to freshly revealed female flowers. So the dye was applied to the tip here of the female flower tepal, and I came back after 24 hours. So it was applied in the morning. I came back after 24 hours, and you can see that it was just sucked inside the plant. So yes, infection can certainly occur through open xylem of the flowers and fruit. Yeah, this result really blew me away. So first, we've shown that a really low concentration of bacteria, perhaps only a few cells, is enough to cause disease. We have shown that infection occurs through the male and female parts of the inflorescence. And our data strongly supports the theory that infection occurs through open xylem. So we can conclude, with this data, we can conclude that protection of the inflorescence from insect visitation would reduce insect-mediated disease transmission. This means that bagging of the inflorescence at emergence in sealed cloth bags and removing the male bell by snapping it off would assist to reduce transmission in the field. Now, if you'd like further information or details on this chapter, susceptibility of the banana inflorescence to blood disease has already been published in Phytopathology. Excuse me. I'll just have a drink of water. All right. So for this chapter, we explore transmission of blood disease in banana. So we first need to determine which parts are infectious. That is, which parts produce sap or ooze that is capable of causing infections. Then we want to understand where the infection causes are and how transmission actually occurs. Can infection occur through the cut surfaces generated during routine pruning and harvesting practices? Do tools actually transmit the bacteria? And can the disease pass from the roots of a diseased plant to the roots of a healthy plant? And can the disease pass from a mother plant to the sucker? So for this section of work, we continue to use the field trial and the potted bananas. It's all the same as what I presented in the previous section. Now, to understand plant-to-plant transmission, we first need to understand where the inoculum comes from or what plant parts are actually infectious. So we collected the sap and ooze from different symptomatic plant parts. We collected it from the male bell, the rachis, the bunch peduncle, the pseudostem, and the fruit. And then I injected that sap and ooze into potted indicator plants. And you can see here that I monitored those plants for symptom expression. So that's the control, that's the symptom expression. And what we found is that all of the symptomatic plants, or the sap and ooze from all of the symptomatic plant parts, is actually infective. You can see here from my table, so for plants, all of the plant parts were infected. Now, for this next experiment, we used field-grown banana plants. Does infection occur through fresh cut surfaces? These are the cuts made to the plant that resemble those made during routine pruning and harvesting activities. So can the bacteria just infect through those cuts? So we spray inoculated the fresh cuts of the petiole, the pseudostem, bunch peduncle, the corm, and the snapped rashes when we removed the bell. We treated Kepok and Cavendish banana plants, and this experiment was replicated and repeated. So we monitored the plants for symptoms of wilt and vascular staining over time. And here are the results at 22 weeks. Now, the important thing is that infection occurred through every single cut surface. So when we cut the petiole, bacteria that was sprayed onto there would cause disease in the whole plant. Same for the other plant parts that we tested. I'm moving on to the next experiment. Now, for this experiment, we used potted plants. We wanted to know if the cultivation practices of pruning and harvesting actually transmit the disease. So specifically, do tools transmit the bacterium? So for this experiment, I stem inoculated some potted banana plants, Kepok and Cavendish, and then I waited for the symptoms to develop. So I waited, I think it was 12 days, and you got the early symptoms of wilt happening. Then I chopped the top off the plant that I was using as inoculum, and I waited for the sap to pull to the top. Once the sap pulled to the top, I used the knife and I inoculated the plants. Now, every plant that I inoculated using a knife dipped in that sap developed blood disease. And both of the strains we tested. The disease is 100% tool transferable, or very highly tool transmissible. So this result actually, I certainly wasn't expecting it to be quite this tool transmissible. Now, in this experiment, we wanted to know if root-to-root transmission occurs. So to answer this question, I grew 2 plants in every pot. And then what I did was I inoculated one of them, and we monitored for symptoms over time. So now transmission occurred from a diseased to a healthy plant, but only in one of the 24 pots that I'd set up for this experiment. We did it in Cavendish, we did it in Kepok. I used 2 different strains. It was repeated and it was replicated. And we only got it in one of the 24 pots. So the results of this experiment really surprised me. This was not at all what I was expecting. When this experiment was done with Ralstonia solanacearum and paired tomato plants. So it was done by somebody else, and it was done MOCO disease. So it was done with Ralstonia solanacearum on bananas. When they did this experiment, the transmission rates were close to 100%. So it was highly transmissible from the root of one plant or the root of a diseased plant to the root of a healthy plant. Very highly transmissible in Ralstonia solanacearum. But there's a difference between the other Ralstonias and blood disease. Now, this difference in pathogenicity might be due to this biological difference. Blood disease lacks flagellum, and the other Ralstonias all have flagellum. Now, flagellum are really important. They're a really important factor when it comes to pathogenicity in soil. So they allow the bacterium to kind of, find its way to an infection caught in the roots. So now that may explain this result here. So blood disease is quite different from the other Ralstonias. So we used the field-grown banana plants to assess if blood disease can pass from the mother plant to the sucker. So we inoculated mother plants and monitored the suckers for disease over time. Now, as you can see in my table here, no disease was observed at the 5-week mark in any of the suckers from the inoculated parent plants. But after 22 weeks, at least one sucker with symptoms was present on each of the parent plants that I had inoculated. So blood disease certainly moves from the parent plant to the sucker, but there's certainly a bit of a time delay here. Now, this is important because most banana plants don't produce seed. We eat the fruit and there's no seed in there. And they're propagated through tissue culture and through shoots or suckers that are that come off the corm of the main mother plant. So people commonly use suckers for replanting and they move them around the country. So this data supports the conclusion that long distance dispersal is through the movement of contaminated planting materials. We found that sap and ooze from symptomatic plants was highly infectious. Cut surfaces act as infection courts, and the disease is highly tool transmissible. Our data as a whole supports the hypothesis that local dispersal of blood disease is through the mechanical transmission of the bacteria on the bodies of insects, birds, bats, and through human activities. And that the transfer of birds, bats, and human activities happens from just diseased to healthy plants. So basically, anything can move the bacteria, can mechanically transfer the bacteria from diseased to healthy banana plants. Now, long distance dispersal is definitely through the movement of infected planting materials. So we conclude that to prevent transmission, so we use this data to conclude that to prevent transmission, we need to protect the inflorescence through bagging. We need to debell by either snapping the bell off or using a fork stick. We need to clean our tools. So tool sanitation is really important. And we need to use clean planting materials. Now, this section has been published in Plant Disease, Transmission of Blood Disease in Banana. So if you'd like further information, this is a great place to have a look. Now we move to the final part of my talk, the diagnostics of banana blood disease. Diagnostic methods are an important component of disease management and eradication strategies. To address the current lack of validated diagnostic assays, we specifically sought to test published assays with rigorous validation parameters. And then we designed and validated a novel real-time PCR assay. And for this body of work, I'd like to acknowledge the significant contribution made by Vivian Rincon-Flores, who has recently published this section in Plant Disease. Now, blood disease caused by Ralstonia syzygiae subspecies celebens causes, also known as the blood disease bacterium, causes banana blood disease. Now, this bacterium has a bipartite genome structure. It's comprised of a chromosome and a megaplasmid. So there are two previously published sets of primers. BDB2400 targets part of the chromosome. BDB121 targets part of the megaplasmid. Now, neither of these primer sets were validated to a particularly high standard when they were initially published. And both were screened on less than 25 isolates of blood disease that originated from a very limited geographic range. Now, Vivian designed the real-time primers and a prime-time probe from a chromosomal phage tail protein coding sequence as a high throughput option. So it targets part of the chromosome. For a diagnostic test to be good, it needs to be validated. But we also need to know what we are validating, which is why positive controls are so important. So they're really important in every molecular diagnostic we do, and they're certainly important in the validation of assays. So here we use reference isolates that originated from Irida Safni's polyphasic taxonomic revision. So she published that as part of her PhD in 2014. And we also used isolates that had been through Kopp's postulates and proven to cause blood disease. So using this confirms the identity. Now, we sought to validate these primers to NATA standards. So we test the assay specifically against BDB isolates from a wide geographic range. And we test that the assay also does not amplify other strains of Ralstonia solanacearum species complex, and that it also does not amplify other species of bacteria from the microbiome of banana. We then look at the limit of detection, and this is the sensitivity of the assay, and it's done through a dilution series. We then look at the repeatability of the assay. So the assay is repeated multiple times, and we ensure that we get the same result every time we do the assay. So reproducibility, now that's done by a different person, a different human being, actually going and doing the assay in the laboratory. And when they bring back the same result, we know it's reproducible. So we checked the ruggedness of the assay by changing the chemistry. So here we changed the TAC. Now, analytical specificity was tested with 174 isolates of blood disease from a wide geographic area. We used the isolates from right across Indonesia that we'd collected. And we found that this BDB2400, this previously published assay, could only detect 136 of these isolates of blood disease out of the 174. Now, which means that if you use this primer pair to identify, well, we got 38 false negatives. So it's just not reliable enough to detect all of the strains of blood disease. So because it failed this first part in the diagnostic assay, we dropped it from we dropped it from further analysis. Now, the 121 assay could detect all isolates of blood disease, and the real-time PCR could also detect all strains of blood disease. What's important is that other Ralstonia or other species in the Ralstonia solanacearum species complex were not amplified, and also the bacterial endophytes were not amplified. So here we check the standard PCR assays for sensitivity and repeatability. So we're looking at the limit of detection or its sensitivity, and this was through the dilution series. And we find that these assays are very, very sensitive, and they were repeated multiple times, and you can see the same result. They're very repeatable. For the real-time PCR, the dilution series or limit of detection confirmed that these assays were very, very sensitive and again, very repeatable. So one of the standard PCRs and the novel real-time assay passed our validation process. So these target different parts of the genome, which increases our confidence in pathogen detection and identification if you use both of them. So we now have accurate, robust, and sensitive diagnostics available to definitively identify banana blood disease. And if you'd like more information on the specifics of this, we have published this part in Plant Disease. So through this 4-part study, we have a greater understanding of the biology and epidemiology of blood disease. Now, this improves the disease management options to reduce grower losses and reduce disease spread. Blood disease is certainly an emerging threat to cultivation of banana in Southeast Asia, Australia, and beyond. Efforts to manage the disease and prevent further spread to disease-free areas are urgently required. And there's still so much work we don't know, so much we don't know about the biology of blood disease. And there's so much research that can actually be done. We can refine disease management. For example, how do we sanitize field knives? What's the role of soil and water in disease transmission? We could screen for host resistance, and we could determine if strains with different biological properties exist. There's just so much we still don't know about this bacteria. Now, if you'd like more information on any of this, we have publications here. I've made them available to the organising committee, so just contact them and they can forward you copies of these 4 papers that I've spoken about today. I'm honoured to be able to present to you today, and thank you very much for listening. Thank you for inviting me.
Thank you, Jane. It's very interesting talk from you. I believe a lot of students get many inspirations from your talk today. It is very interesting that understanding the biology and epidemiology like that you did. It's really important as a basic for disease management. As I mentioned before, before we head to discussion section, I'll have a short quiz for participants, which 2 participants that can answer first, we will determine as a winner, and from Plant Protection Department will send you a special gift, and we will have 2 winners from Zoom and YouTube. Probably for the organizing committee, can you please share the question, and participant can answer the question through the chat. First question will be, where was banana blood disease firstly reported in Indonesia? For participants, please, you can answer the question through the chat. Okay, Pak Gianto, Mas Hari. Okay, come in. Firstly, come first. I think for the organizing committee couldn't participate in this quiz.
Oh, I'm sorry. No. Oh.
Okay, so the organizing committee will choose the winner. Okay, a lot of answer. Quick. Still.
Pak Gianto nggak boleh jawab.
I don't know the rule.
Oh, sorry. Okay. We will.
I'm a student
Move.
actually. In the seminar, I'm a student attendant.
Okay, move to second question, please. What percentage of plants became diseased from tool transmission? So Jane conducted several disease transmission. So our question is, what percentage of plants became diseased from tool transmission? Okay, 1, 2, 3, 4. Oh my God. It indicates everyone listen very carefully. Still one more. For participants on YouTube also can answer the question, and we also will choose 2 winners from YouTube participants. So it will be, there are 4 winners, 2 from YouTube and 2 from Zoom. And the organizing committee will contact the winner. Okay, thank you so much, Jen. And okay, now we will move to the discussion section. I think there are 2 questions. The first one from Asti from IPB. The question is, Kapok and Cavendish are considered very susceptible to BBD. They have different genome type. Kapok is ABB, while Cavendish is AAA. Do you think susceptibility or resistance of banana cultivars has no relationship with genome type? Just for your information, bananas with B genome is more resistant than those with A genome in the case of banana bunchy top virus.
Yeah. At the moment, it doesn't look. We're unsure at the moment. We haven't found a plant with resistance as such to banana blood disease. So, in fact, I think Abaca. We even found banana blood disease to infect Abaca. We found that in the field. So if tool transmission occurs, for example, then the plant can certainly become, the pathogen can certainly get into the plant xylem and cause disease. But there are a couple of plants that don't have bells in Indonesia, and they become, they're much less susceptible. They avoid infection, so to speak. So you've got, I think it's Pisang Puju and Pisang Tanduk. Yeah. Pisang Puju, and yeah. So the lack of the bell reduces insect transmission to that infection cord. But there's not actually true resistance, because you can, if you hit them with a knife, they will become sick, for example.
Thank you, Jen.
So it would be nice to find some resistance somewhere, but I'm not sure where it exists at the moment. Oh, Ivan, Gauman, who did research more than about 100 years ago. So he actually looked at about 100 different varieties of bananas in his field trials in Sulawesi, and he found that all of those bananas eventually succumbed to disease.
Thank you. Lasti, do you have any comments or further questions?
No, I think that's clear. Thanks, Jane.
Thanks,
My
Jen.
pleasure.
Okay, for second question came from Gianto, which is a student in this talk. So, Pak Gianto, dear Jane, very interesting information. My question are, 1, what kind of inoculum you use for inoculation, in bracket, cultured in laboratory or from the set of symptomatic plant? And the second question, do you have any experience to keep the virulence of bacteria after culturing in laboratory? And then the third one is how...
You are.
Okay, Okay, probably just first question first.
Yeah, we'll have to do one at a time.
Okay.
They're fabulous questions. All of them are fabulous questions. So
Okay.
now the first question was
Okay. Thank you. We will summarize in a PowerPoint slide, Jane, so you can
okay.
question.
Okay, interesting. Question one. What kind of inoculation do you use for inoculation? Okay, so the inoculation was different depending on the experiment. So most of the field experiments that I did, I made inoculum on CPG media, and I just diluted that to 10 to the 7 colony forming units per mil, and then spray inoculated that on the plant. So every time I checked the concentration of the inoculum, that it wasn't too high. And I checked that the inoculum was viable afterwards. So that was grown on culture media. And I think the cultures were, because the bacteria grows a little bit slowly, I think the cultures might have been 2 days old, not 1 day old. And we did use, so in the experiment where we tested the which plant parts were infectious, so we collected the sap and ooze from all of the different plant parts. So from the male bell, so the male bell produced, it was oozing bacteria about, I think it was 12 to 14 days after I had initially inoculated the plant, and it was raining at the time, so there was a lot of high humidity, but the male bells produced sap. So I collected that sap from the plant and used that as inoculum. And then other plants, I collected the sap from symptomatic plant parts. So those plants had been inoculated about 5 weeks beforehand. So the disease was fairly fresh in all of the plant parts. So you can use both as inoculum, but they were for different purposes. It depends on the research question. Now, yes, okay. Yeah, the virulence of the bacteria after culturing. So with Ralstonia, there's a couple of things that go on with the Ralstonia. So firstly, they don't like it. They don't like being cold. So you can't fridge store isolates at any point in time, or your plant material if you want to isolate from them. So other bacteria you can get away with storing them, keeping them cool for a period of time, but not Ralstonia. They really don't like it. So to keep the virulence of the bacteria, one thing that I did was you need to shorten, not keep them in culture too long. So I isolated them on TZC media, or media amended CPG and tetrazolium chloride media. And then I subcultured them onto CPG media. And as soon as they were in pure culture, I would obtain a streak. I'd make a streak, a plate full of the bacterium, and then I'd store that in a little tube of sterile water. It's super, super basic and super, super easy. Little tubes of sterile water. So you put the inoculum in there, and the bacterium seems to survive in there for, I didn't actually lose any cultures. So I'd say anything from 2 years to 10 years. Like, I'm not sure the length of time, but certainly that's what I used. So for my whole PhD, I stored the bacteria just in a ml of water in a little baby sterile tube. And then when I went to, and it kind of keeps the bacteria in a happy state, or suspended. And then when I went to make inoculum, I would always just bring it straight out of that sterile culture and make inoculum, make plates on, I think I did TZC to check that it was pure, and then onto CPG, and then used that directly for very fresh to make inoculum. So the inoculum was always very freshly made from stored water. So I never kept the bacteria growing on plates for any great length of time, not more than a couple of weeks, because you do run the risk of the bacterium losing virulence. And this is a super common problem with Ralstonias. Sorry, I'm talking a lot, but now how the role of insect pollinators or pests in the disease. Sorry, I need to minimize.
It's spread.
Oh, in the disease spread. Yep. So yes. Insect pollinators, so basically anything that visits the inflorescence or any part of the plant that is symptomatic. So for instance, if a grower cuts the bell off a plant or harvests a banana pseudostem and that has blood disease in it, and there is, the sap is coming up the pseudostem. So that sap, all of the sap from symptomatic freshly cut plant parts is actually infectious. So if an insect lands on there or anything lands on a cut pseudostem with symptoms and fresh contaminated sap, and then it flies to any part of the banana inflorescence. So if it flies to the parts of the male bell with those open xylem, then the bacteria is just likely to get sucked inside and initiate an infection. And the same, so if it lands on the flowers, it can also, it's less likely, but it can actually cause infection through that way. So insects, birds, bats, anything, tools, humans, hands, anything can transfer the bacteria from a diseased symptomatic plant to a healthy plant, to an infection port of a healthy plant.
Thank you so much, Jen. It's very clear answers. Okay, for participants that we are waiting the question from, the third question came from Widi Nugraheni. It came from YouTube.
Oh, yeah. Mm-hmm. Sumba and its neighborhood farmers tend to use them as animal feed. No, I don't believe that I think that blood disease doesn't hurt. There's no evidence to suggest that the disease that causes disease in banana plants can harm humans or animals. It's just that the banana fruit looks so awful that us humans certainly wouldn't want to eat it. And I'm sure it doesn't taste very good. I didn't actually try it, but so there's been no research to show that it causes a problem, a health issue for animals or humans. I think it would be, I think it's fairly specific for blood disease, for bananas, sorry.
Yeah, true.
Terrific question though.
Yeah, it is true. It's very rare that plant disease can cause or harms to humans. Okay, I believe that there are a lot of bacteriologists or students who works in the plant bacteria field. Probably atau ibu-ibu, bapak-bapak kalau malu untuk bertanya dalam bahasa Inggris, saya akan membantu untuk menerjemahkan. Jadi jangan malu-malu apabila ada pertanyaan yang ingin disampaikan ke Jen. So I just try to explain. If they are shy to ask a question in English, I will have to translate.
That's okay.
Yes.
Okay.
I am so sorry that I don't speak Indonesian.
No, that's fine. Totally fine. Okay.
I want to from France.
Okay.
North
Okay,
Sulawesi, Komda PFI Sulawesi Utara.
Oh, wow. Yeah, but
No.
Franz came from North Sulawesi, Jane.
Oh, yeah.
Yeah, I know. Formerly, we have done isolation of Ralstonia from all of insect visiting
Yeah.
in flowers
Yeah.
to banana inflorescence.
Yeah.
And our result, all of the insect have carry
Yeah.
Ralstonia. It's very amazing. All of the insect visiting banana flower is carry this bacteria. And then I want to know about virulent or not virulent Ralstonia. How about that?
Oh, if I'm not sure of your question. So but I think I can try and answer. So now
Yes.
when we looked at we did a dilution series and we injected the potted plants with blood disease from 10 to the 10 colony forming units down to really super dilute 10 to the 2 colony forming units per mil. And when we even injected the plants with 10 to the 2 colony forming units per mil, we found that all of the plants developed symptoms. So basically only one or a few bacteria just need to, that are viable, need to land on open xylem, and it will cause or get into the xylem of the plant, and it will cause a full-blown infection. So eventually the disease will be systemic in the plant and it will cause full blood disease. So it's a very, very virulent and aggressive pathogen. And so all of those insects that you looked at are likely to be able to move the bacteria from a diseased plant to a healthy plant.
Sorry. I mean, if you or in laboratory to isolation of this bacteria, use TCC, sodium chloride, as a media for isolation this bacteria. Yes.
I only used one type of media for the isolation of the bacteria. I'm sure there are other types of media that you can isolate the bacteria on, but I just used it's Kelman's. It's a very old recipe from, I think Kelman published this recipe in 1968 or something. And it's Kelman's TZC recipe, and all it is a CPG or a very basic media with a little bit of tetrazolium chloride in it. And that's the only media that I experimented with. So I didn't try other medias. Other medias may work, and perhaps somebody else in the audience has some more information on what works for them.
In your result of isolation, you didn't found a virulent colony in your media.
A virulent colony. Is that what you're asking for?
A virulent.
Yeah. Okay. Yes. Yeah. Okay. So that hasn't been published. Yes. Now I understand your question. Okay. So in Ralstonia solanacearum species complex, normally when you grow the medium, when you grow the bacteria on TZC medium, you get fluidal and non-fluidal colonies, and the non-fluidal colonies or the mutants, when you inject those into a plant, they can't cause disease. So they can drop their virulence, but you can see it in the culture media. Now, when I looked at blood disease. Blood disease is very, very slow growing anyway. And they kind of, and so you can't really see that avirulence on the plate as such. But I did inject one isolate that I was working with and into a plant, and it didn't cause disease. So it had certainly lost its virulence. But you can't, it doesn't really, you can't, I didn't notice there was a massive difference on the plate, but it certainly does lose its virulence like other Ralstonias. It's very common in that group of bacteria. Does that make sense?
Yes, terima kasih.
Yeah.
Thank you very much.
Thank you, Pak Fran,
got there in
for
the
your
end.
question.
So there are, and you have to really look after your isolates very, very carefully to make sure that they maintain their virulence, which is what I was talking about, storing them under 1 ml of sterile water. That really helps them maintain their virulence.
Okay. Good, good.
Thanks, Pak Frans. Is there any question from participants, either from Zoom or YouTube, or probably the alumni that has worked with the plant pathogenic bacteria? Or you can just type the question and we will read the question.
While waiting, may I ask one more question?
Sure, Bu Asti. Very welcome.
Again, you mentioned,
thank you so much.
You mentioned earlier that this disease is an exotic disease for Australia, right?
Yeah.
So from your research, what will you recommend for the Australian government, to especially the detection technique that you will recommend to check for the association of this bacteria to banana plant materials, for example?
Yeah. So now we published a lot of that in the last paper, and I would recommend. So in terms of, so if we get a disease symptom in Australia, we'd first have to determine, because we also don't have MOCO and we don't have Xanthomonas wilt, so we haven't got any of those bacterial wilts of banana in Australia. So we'd have to determine if it was Xanthomonas wilt first, or MOCO, or caused by a Ralstonia solanacearum species complex. But what I would recommend, if we suspect that it's caused by blood disease, is I'd recommend to use the 1-to-1 primers and Vivian's real-time PCR. So normally in Australia, we have to use more than one diagnostic method to initiate a response as such. So we have to really, really prove that we've definitely got an exotic pathogen. We have to do a lot of work. But what I would recommend is that I'd actually recommend that at the moment that there's a lot of scope for more research into banana blood disease, because relatively well, there's more research into the bacterial wilts in general. And from the Australian perspective, MOCO is very high profile on the radar of Australia, but MOCO is actually still a fair way away from Australia, and we probably know a bit more about MOCO in some respects than blood disease. But yeah, so I'd recommend more research so we can understand the biology of the organism. We really, it would be really great to work out how to manage the disease so that farmers could eradicate in Indonesia or manage the disease a little bit better. You know, how they clean their tools. In South America, for instance, the farmers actually have like kind of like a pouch that they store their machetes in, and they have that on there. They normally have 2 machetes, one on either side, so they've got 2 pouches, and the pouches contain like, it's not bleach, but it's a solution that sterilizes the knives, or it gets rid of the Ralstonia, the bacteria from the knives. And so they're so used to doing it because they've got very, very big plantations there. And each time they make a cut to a plant, they then put the knife back into a pouch, and then they grab the other knife and make the next cut to a plant and put it back in a pouch. And that's how they're so used to doing it, because Moko is such a problem over there in the massive Cavendish plantations that they have. And so research to do something like that in Indonesia that's that is potentially would be culturally appropriate for the farmers, something that they might uptake, would be really, is really the next step. It's really, yeah, it's a very valuable thing to do. Yep. Sorry, I probably got a bit too.
Oh, no. Thanks, Jen.
Thanks, Jen. It's also like a knowledge for us how the biosecurity works in Australia and how they prevent the emerging disease or new disease to enter to Australia. It's very clear explanation. Thank you so much. Okay, probably I still give a chance for the participants who will ask Jane about the banana plant disease, because it's very interesting. Jane has conducted the research from the field to the laboratory, and it's really, really good. Okay.
Very fundamental research, very applied research, which, yeah, similar to your research in Brisbane.
Yes.
Very applied.
Yes, we shared a lot our experience during the field experiments. Okay, for the students from Zoom or from YouTube, are there still any questions to Jen? You can ask in either way. I mean, in Bahasa or in English. No? Okay, Jen, thank you so much. I think before I close the call, probably I can summarize 3 main points. Like, it is really important, like we got a very good lessons from you, that understanding of biological character of pathogen and understand how the epidemiology of the pathogen are really important to control the disease. And also we know how to some of the steps to prevent the transmission of the disease in the fields, with protect the inflorescence from the insect, or with the tool sanitations, and use the clean planting materials, and also how the important to get a very robust detection technique. Thank you so much, Jen. Thank you so much, and I will return the seminar to MC. Mas Hari.
Thank you, Bu Ita. And thank you, Jen, once again for the inspiring talk in today's lecture, and Bu Ita for summarizing the presentation also. We will now have a certificate of appreciation to our excellent guest lecturer and, of course, our chairman. And for this opportunity, I would like to invite once again Bu Asti for the certificate of appreciation. Please, Bu Asti.
Okay, thanks, Hari. So Jane, very interesting and excellent research. Congratulations for such big and great achievement in your research. And also, more importantly for us here now, Thanks so much for sharing with us all of the story about the banana blood disease. So please receive this certificate as the token of appreciation from us for your contribution to our guest lecture program. Thank you, Jane. This is the certificate for you.
Thank you very much. And I'm just so grateful for the opportunity to share the research that I've done with you and your audience. So I'm very, very grateful for the opportunity that you've provided me today. Thank you very much.
All right. I hope
it's my total honor and pleasure. I'm sorry.
Yeah. I hope we can meet each other sometimes in the future in person, Jane, not
Yes.
virtually.
Yes, that would be very nice.
Yeah. Okay, kemudian ya, also certificate for Sari Nurulita as PMSE for becoming our moderator for today's lectures. Bu Ita, terima kasih banyak sudah memandu acara guest lecture ini dengan sangat baik. Mudah-mudahan cepat tertular oleh Jane, cepat lulus dan bergabung kembali dengan Departemen Proteksi Tanaman. Terima kasih.
Terima kasih.
Okay, Hari, I think it's back to you.
Thank you, Bu Asti, for the certificate of appreciation to our guest lecturer and also our chairman today. And now, this is the moment of truth, the announcement that we have been waiting for, the winner of previous quiz. From Zoom, we have Ibu Sri Nur Aminah and also Ibu Herlina Raharja Putri. I know the second one. And then from YouTube, we have Pak Andi Suryadi and also Ibu Widi Nugraheni. Oh, Ibu Widi Nugraheni is the banana team. It's from Wageningen University, from Amsterdam. I mean, the University of Amsterdam in the Netherlands. For all of the winner, please contact Ibu Andika Septiana in this number to confirm your address, where the present will be sent directly to your address by our team. And ladies and gentlemen, we are now in the end of our session today. Please do not forget to fill in the attendance through the link that has been shared in the chat room. Especially for those who are enrolled in plant pathogenic bacteria course, don't forget to fill in the attendance. On behalf of the organizing committee, I would like to thank you all for joining today's guest lecture. And we do hope that you are enjoying the lecture, and also we look forward to seeing you all at our next guest lecture series. I wish you all a pleasant day. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you.
Thank you.
Terima kasih
Thank you so
semua.
much, Jen.
Oh,
Thank you,
my
Jane.
pleasure. I'm very thanks for having me.
Thank you. Bye.
Hari, mana Hari? Makasih ya, Har.
Makasih
Hadir,
Mas
Bu,
Hari.
hadir. Sama-sama.
Bagus loh gitu tadi ini.
Terasa di pesawat saya. Terasa di pesawat Jakarta Amsterdam, Har.
Sengaja Bu itu Bu.
Ya kan ya?
Heni,
Pak Nadir
Heni
ya.
tadi dari UQ, University Amsterdam.
Oh iya.
Iya.
Oh, di sana ada tim banana itu di sistem.
Ada Bu, yang TR Res 4 itu.
Oh, yang fusarium. Oke.
Ya.
Masih ada terus ya fusarium ya, penelitian fusarium ya.
Masih. Mereka koleksi banyak plasma nutfah dari Indonesia.
Apa fusariumnya atau banananya?
Banananya, Bang.
Oh, oke. Terus di mana ininya? Greenhouse-nya?
Greenhouse-nya di
mana?
Di Wageningen.
Oh, di Wageningen.
Bawah tanah ya Mas ya biasanya.
Sepertinya iya, Pak. Sepertinya iya.
Jepang juga begitu, punya koleksi bawah tanah itu rumput-rumputan seluruh dunia juga dikoleksi. Kita aja yang nggak open.
Itu gimana bawa plasma nutfahnya sampai ke Amsterdam?
Mereka ada kerjasama sih dengan LIPI ya kemarin. Jadi dari LIPI yang menguruskan.
Sekarang udah nggak boleh kayaknya ya?
Seharusnya dari dulu pun nggak boleh.
Dulu banget itu koleksinya sebelum
harusnya nggak boleh.
2000-an, tahun berapa udah?
Iya, dari dulu boleh, terus terakhir-akhir kan bukan nggak boleh tapi dipersulit ininya